Today, Group 14 will present about factor P, also known as piperidine, focusing on its structure, function, and clinical relevance. First, what is factor P? Factor P is the positive regulator of the alternative complement pathway. Unlike the classical and lectin pathways, the alternative pathway can be activated spontaneously. Factor P plays a key role by strengthening and sustaining this activation. Next, let's look at where factor P is produced. As shown in a slide, it is mainly produced by neutrophils, monocytes, and a subset of T cells. When these immune cells migrate to sites of infection, they release factor P locally, helping the complement system act efficiently exactly where it is needed. Now, moving on to the structure of factor P. Piperidine exists as dimers, trimers, and tetramers, and each molecule is composed of repeated domains called thrombosponding type 1 repeats, or TSRs. These TSR units allow factor P to bind to complement components and stabilize the convertase complexes. Next, we explain the functional role of factor P. In the alternative pathway, the central enzyme complex is C3 convertase, known as C3b. Factor P binds to this complex and stabilizes it, preventing it from breaking down too quickly. By stabilizing C3 convertase, factor P increases the rate of C3 cleavage, leading to the production of more C3a and C3b, and thereby amplifying complement activation. As C3b accumulates, C5 convertase is formed, which then triggers the assembly of the membrane attack complex, MAC. Because factor P supports the formation and persistence of both C3 and C5 convertase, it plays an essential role from the early to late stages of the complement cascade. Factor P also helps localize complement activation specifically to the surface of pathogens, preventing unnecessary damage to host tissues. In addition, it enhances the function of immune cells by supporting opsonization and inflammation. Next, we'll discuss factor P deficiency. Piperidine deficiency is a rare X-linked disorder, and individuals with this deficiency are highly susceptible to infections by Neisseria meningitis, the bacterium that causes meningitis. Because the alternative pathway cannot function effectively, the immune system struggles to eliminate these pathogens early in infection. Finally, factor P can be involved in disease not only when it is deficient, but also when it is excessive. When piperidine levels are too low, alternative pathway activation is impaired, leading to recurrent infections. Conversely, when piperidine levels are too high, complement activation can become dysregulated, contributing to excessive inflammation and tissue damage. To summarize, factor P is the only passive. This concludes our presentation. Thank you for listening.